Hi there. Today we're going to take a quick look at cell communication. So, an overview of cell communication. So, when we're looking at cell communication, we are looking at how cells send messages and receive messages and then respond to those and how they cause a response in the cell. And an example of that is the fight or flight response, which is triggered by a signaling molecule called epinephrine. So, in that fight or flight scenario, that hormone is released, and cellular response is going to help that prey get away from the predator. And we'll talk more about epinephrine a little bit later on when we talk about hormones and the endocrine system. So, microbes are, when we look at microbes and bacteria, like we saw in the video on quorum sensing, the microbes have a role in that signaling process. And from that, we can see the evolution of life on Earth going from prokaryotes to eukaryotes and multicellularity coming out of that cell signaling process. So when we look at cell signaling, it involves a signal transduction pathway. And a signal transduction pathway is a series of steps by which a signal on a cell surface is converted into a specific cellular response. And an example of that is in yeast. When you've got yeast that are mating, you've got two different types of yeast, and each of those have message molecules that they secrete, which are then picked up by a receptor protein on the opposite cell. And so when they each receive that signaling molecule, they then will mate with each other and create a new cell. So that's an example of those signaling molecules. And we can kind of see similarities between ancestral signaling molecules and eukaryotic cells today in multicellularity, again, like we saw in the quorum sensing video. When we talk about cell signaling, we've got local signaling and we've got long distance signaling. Local signaling is going to be when you're sending out a signal and it doesn't go very far to the cells that are in the local area. And then long distance signaling is when a message goes a long distance. So think of it like you want to throw a party and you want to invite all of the neighbors. And so you go out and you put a flyer in all of your mailboxes of your neighbors, and then they know that you're having a party and that they're invited. So that would be local signaling. You're sending those messages out just to the neighborhood. But let's say you also want to invite your grandma who lives in Florida, a long distance away. You're going to have to stick that letter or an invitation in the mail, and then that's going to go to grandma, and she can then decide if she wants to come to your party or not. So that would be long distance signaling because it involves the postal service carrying the message somewhere else. So in our cells, in our body, we've got both local and long distance signaling. When we look at how cells communicate, sometimes the communication is through connections of the cytoplasm. And we can see that in cell junctions. So gap junctions we would see in animal cells and plasmodesmata in plant cells where the cytoplasm is actually directly connected. So there's gaps in the plasma membrane that allows signaling molecules to transfer directly from one cell to another. We can also see cell communication when we've got receptor proteins and proteins on adjacent cells that are connecting together. And so we see that in cell-to-cell recognition. And you typically see that when you're looking at cells that are of the same tissue type. So those molecules on the surface of the cells are helping with cell-to-cell recognition. And when we look at those local signals, a message that's sent out to cells that are in the nearby vicinity, those are called local regulators. So a local regulator is going to be a messenger molecule that only travels a short distance. So like that, putting the messages in the mailbox of your neighbors, the signal only goes out to a short distance. If a signal molecule is going a long distance, then those molecules are called hormones. So hormones would be signaling molecules that travel through the circulatory system and go a long distance. When they go through the circulatory system, they're going to be picked up by very specific receptor molecules on certain cells. So even though they go throughout the entire body, only the cells that have the receptor site for that particular hormone will attach on and respond to that hormone in those cells. If we look at local signaling, paracrine signaling is an example of a local signal. And so you can see the cell there in the middle is secreting a local regulator, and it's going to the nearby cells, and then those will respond to that signaling message. Synaptic signaling is another example of local signaling. Those two nerve cells are very close together, and one cell will send a message, a chemical message, across the synapse, or that space between the neurons, and then the other neuron is going to pick up that message, and that's going to trigger a response in the next cell. So those are two examples of local signaling. And then long distance signaling would be when you've got an endocrine cell secreting a hormone into the bloodstream, and that goes throughout the entire body, and then it's picked up by a specific target cell that has receptor proteins that will receive that message. So looking at cell signaling and signal transduction pathways, there are 3 steps in a signal transduction pathway. We've got reception, transduction, and response. And so this diagram kind of sums that up. So reception would be your first step when the signaling molecule attaches onto the receptor. And then that's going to cause a series of steps within the cell. So a signal transduction pathway is going to involve kind of like knocking over dominoes. One knocks over the next, which knocks over the next, and so on and so forth. That transduction is that series of steps that occur as a result of that receptor protein attaching on, or the signaling molecule attaching onto the receptor protein. And then in the end, it's going to cause something to happen inside the cell, and that is your response. So your cellular response is that third step. So signal transduction pathway involves reception, transduction, and response. That's kind of an overview of a signal transduction pathway. If we look at each of those steps in a little bit more detail, we can start with reception. And with reception, you're looking at a signaling molecule. That signal molecule is often called a ligand, and that's going to be very specific to a receptor protein that it will attach onto. When the ligand attaches onto the receptor protein, it will cause that receptor protein to change shape, which will then trigger something inside of the cell to happen. Most signal receptors are going to be plasma membrane proteins, so embedded in the plasma membrane. And then because most of those are water-soluble signaling molecules, the receptor protein has to be on the outside of the cell membrane so that it can attach onto the signaling molecule, which will then cause something inside the cell to occur. And there are 3 types of membrane receptor proteins. So we're going to talk about each of those 3. We've got G protein coupled receptors, receptor tyrosine kinases, and ion-gated channels or receptors. And so if we look at the G protein coupled receptors first, they're going to be your largest family of proteins. And what will happen is the G protein coupled receptor will have the signaling molecule attach onto it, which is then going to activate the G protein. So normally the G protein is in an inactive form. And then when the signaling molecule attaches on, so here in purple is your G protein coupled receptor. When the signaling molecule attaches onto it, the G protein coupled receptor changes shape, which will then activate the G protein. And that will require energy. So in this diagram, it's showing you GTP, which is very similar to ATP, a source of cellular energy. That GTP is activating that G protein, which will then cause that G protein to shift, and it will move over and activate another enzyme in that signal transduction pathway. And then that G protein will reset and become inactivated again. But the active enzyme will then cause something else inside the cell to occur. So it will trigger a cellular response. Then the whole thing resets and is ready to receive the next signaling molecule. So the G protein coupled receptor activates the G protein, which activates an enzyme, which eventually really will lead to a cellular response. And here's just another diagram showing you that same thing. So when an appropriate signaling molecule binds to the extracellular side of the receptor is activated and changes shape. Its cytoplasmic side then binds and activates a G protein. The activated G protein carries a GTP molecule. Then the activated G protein leaves the receptor, diffuses along the membrane, and then binds to an enzyme, altering the enzyme's shape and activity. Once activated, the enzyme can trigger the next step, leading to a cellular response. Binding of the signaling molecule is reversible. The activating change in the GPCR, as well as the changes in the G protein and enzyme, are only temporary, and these molecules soon become available for reuse. So basically, it resets. Okay. The next one is a receptor tyrosine kinase, and those are also going to be membrane receptors that will attach phosphates to the tyrosines. And we talked about this in our energy unit. Anytime you attach a phosphate group onto a molecule, you're basically activating it or energizing it. And so in this scenario, the signaling molecule will cause the receptor tyrosine kinases to phosphorylate and then become active. And so it's going to activate multiple tyrosines, which each one can then trigger multiple cellular signal transduction pathways at the same time. So it can lead to multiple responses within the cell. So if we look at this diagram here, here are your receptor tyrosine kinases in an inactive form. And then what will happen is when the signaling molecules bind to that, they combine to form a dimer, and then ATP will phosphorylate these tyrosines, so they become phosphorylated with the phosphate groups from ATP. The energy is transferred from the ATP to each of those tyrosines. And so all 6 of them are phosphorylated. So all 6 of them can then trigger a cellular response. So the difference between this and the G protein is this can lead to multiple cellular responses at the same time. The G protein coupled receptor typically is just going to lead to a single cellular response. But this one, each activated or phosphorylated tyrosine can activate a cellular response in the cell. So multiple signal transduction pathways can occur in those cells as a response to the signaling molecules. And then the last one is a ligand-gated ion channel. And when the ligand binds to that receptor protein, it opens up an ion channel. And the signaling molecule could open up an ion channel for a bunch of different types of ions. And when we talk about the nervous system a little bit later, we will talk about how those ion channels work in sending signal messages through the neuron. but it could be a sodium channel or a calcium channel, positively or negatively charged ion channels. It's going to open that, and it's going to allow those ions to diffuse through that channel from high concentration to low concentration. So here's your signaling molecule. Normally that ligand-gated ion channel is closed, but when the signaling molecule attaches onto it, it opens that ion channel, and ions can diffuse through, again, from high concentration to low concentration. And then the signaling molecule will come out, and the gate closes again and resets and waits for the next signaling molecule to come in. And so those are your ion-gated channels. All of those three are your typical signaling molecules or receptor proteins that you would find in the membrane. But sometimes those membrane proteins are on the inside, those signaling receptor proteins are on the inside of the cell. And so those are going to be intracellular receptors. And that's going to be on the inside because the signaling molecule in those cases are going to be able to pass through the cell membrane. So they are either small or they are hydrophobic chemical messengers. And so they can easily pass across the cell membrane, and then they're going to activate the receptor, which is on the inside of the cell. And steroid hormones would be examples of intracellular receptors. So in this diagram, you can see up here, you've got testosterone, which is a steroid hormone. And so the receptor protein is on the inside of the cell. And so the hormone will diffuse across the cell membrane and then attach onto the receptor protein. And then that's going to cause your transduction inside of the cell. And in this particular case, we are looking at protein synthesis being initiated. And so the receptor is on the inside of the cell because the signal can pass across that cell membrane. And so those are intracellular receptors when it's on the inside of the cell. So that's the signal part of it. Now looking next at transduction, we are looking at typically a series of steps that are occurring. So again, just like dominoes not being knocked over, one domino knocks over the next one, which knocks over the next one. So multiple steps in that pathway. Sometimes those steps can amplify a signal. And so one molecule could trigger two molecules or activate two molecules, which activate more and more. So oftentimes you're going to get an amplification of signals as well. So that would be your transduction pathway in your cells as a result of those receptor proteins receiving that signal. When we look at transduction, it often involves protein kinases, and protein kinases will transfer phosphate groups from ATP to a protein in a process called phosphorylation. And oftentimes you'll see multiple protein kinases being activated in phosphorylation. And then dephosphorylation is when the phosphate group is removed from the protein, and there is an enzyme called protein phosphatase that removes those phosphate groups. So there's phosphorylation and dephosphorylation involved in those transduction pathways. So this would be one example of a transduction pathway, which is called a phosphorylation cascade. And so in a phosphorylation cascade, what happens is a signal molecule touches onto the receptor, which then will activate an enzyme, which then will activate a protein kinase, which will then activate another protein kinase, which activates another protein kinase, which then activates a protein, which leads to a cellular response. And that's called a phosphorylation cascade when you've got a whole bunch of protein kinases being activated and then a series of reactions leading to an activation of a protein. And so a phosphorylation cascade is a step-by-step, again, just like those dominoes falling over, step-by-step procedure, which leads to that cellular response. Sometimes those phosphorylation cascades are also going to amplify and activate multiple proteins, which then leads to an amplification of a signal. Sometimes those transduction pathways involve other molecules besides proteins. And in those cases, we are looking at second messengers. So the ligand is your first messenger, and a second messenger is going to be a small non-protein water-soluble molecule or ion that spreads throughout the cell by diffusion. And those, because they are not proteins, they are considered second messengers. And some common second messengers are cyclical AMP or CAMP. And there is adenylene cyclase, which is an enzyme that will convert ATP into cyclical AMP or CAMP in response to a cellular signal. So that CAMP is an example of a second messenger. Another common second messenger is calcium ions as well. But here, this diagram kind of shows you that CAMP as a second messenger. So again, your first messenger is your signaling molecule. And in this example, we're looking at a G protein coupled receptor, activates the G protein, which activates adenylene cyclase, which then converts ATP into CAMP, which that then activates the protein kinase, which leads to a cellular response. So this step in that metabolic pathway involves a molecule that is not a protein and therefore a second messenger. Here's just another diagram showing you the same thing. And then calcium ions and IP3 are other examples of second messengers. So you might see some of those in diagrams. Just know that those are also second messengers. And so I'm going to skip that video real quick. So if we look at this diagram here, you've got a signaling molecule activating a G protein, which then activates PIP2, which then is converting that into IP3, which is a second messenger that then activates an ion-gated channel, which allows calcium to diffuse into the cell into the cytoplasm, which then activates various proteins, which then lead to a cellular response. So that IP3 here and the calcium ion down there, those are both examples of second messengers. Let's take a quick look at a little animation that kind of sums up this information.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
Very nice summary of those signal transduction pathways there. And then finally, we're looking at a cellular response. So the cellular response is your output, what the end product is of that signal transduction pathway. And so typically, you're going to have either a nuclear response or a cytoplasmic response, which means something's either going to happen in the nucleus or it's going to happen in the cytoplasm. And if it happens in the nucleus, typically you're looking at the initiation or the starting of protein synthesis. So it will usually involve the activation of a transcription factor, which then causes protein synthesis. So this diagram kind of shows that nuclear response. So a growth factor would be an example of a hormone that is attaching onto that receptor protein, causes a phosphorylation cascade, which then activates a transcription factor, and then transcription starts and protein synthesis begins. So that would be an example of a nuclear response. And then other times you're looking at instead of starting protein synthesis, you are activating enzymes that are already in the cell. And so here we're seeing a reception causing a series of events to happen, a phosphorylation cascade, which is then activating a certain enzyme within the cell. So it could be that you're activating a protein, it could be, or an enzyme, it could be that you are causing proteins or structures to be produced that could change the shape of the cell. There's lots of things that could happen in a cytoplasmic response. So in that video, it talked about changing the structure or the function of proteins or things inside of the cell. And so that would be a cellular cytoplasmic response as opposed to a nuclear response. So it could affect the overall behavior of a cell, like changing its shape. So for example, in those yeast cells that we saw earlier, when they're mating, oftentimes they will create shmoos. So a shmoo is a projection from that yeast. And so the mating factor attaches on and it leads to a transduction pathway that then causes the formation of microfilaments. Those microfilaments move and they cause the cell membrane to create an outbudding or a shmoo, and it's basically changing the shape of those mating yeast. And so that would be an example of a cytoplasmic response. It's changing the structure and the function of the cell through the cytoplasm. And then the response can be fine-tuned in several different ways. So there are 4 aspects of fine-tuning to consider. There's amplification of a signal and thus amplification of the response. There's specificity of the response. There's overall efficiency of the response enhanced by scaffolding proteins, and there's termination of the signal. So when we amplify a signal, the enzyme cascades are going to amplify. So each step along the way is going to activate multiple enzymes or products, and then each of those will activate additional proteins and so on and so forth, leading to lots and lots of enzymes being activated in the end. So amplifying those signals. So each domino is knocking over multiple dominoes, which then knock over multiple dominoes, so you're amplifying the signal. And then when we're looking at specificity of cell signaling, there are different kinds of cells that would have different proteins and they're going to respond in different ways. So they will allow the cells to detect and respond to different signals. So if we look at these diagrams here, this first one, you're looking at that signaling molecule causing a certain response in one cell, But in a different cell, that one same receptor protein is going to activate two responses in the cell, two different responses. So different cells can receive the same signaling molecule and respond in different ways. And then here we're looking at another cell that has that same signaling receptor protein, but when it receives another signal in another receptor protein, it leads to the activation of both of those and leads to a cellular response. So now a different cellular response from that same signal. And then in another cell, you've got a different receptor protein receiving the same signal, but then leading to a completely different response. So different cells are going to respond to those same signaling molecules in different ways. And so that's what we're looking at when we're talking about specificity of the cell signaling. And then if we look at signaling efficiency, there are scaffolding proteins that will kind of allow those proteins to align so that they react faster. So scaffolding proteins will increase the signal transduction efficiency because they're grouping together different proteins involved in the same pathway to make them more efficient. So basically they're kind of lining it up so that they can react efficiently and quickly. And so that's going to enhance that signal transduction pathway. And then termination of the signal, we can inactivate those ligands, and then that would then slow down or stop the signal. So unbound receptors would revert to an inactive state. So inactivation mechanisms can turn off that signaling transduction pathway. And if the ligand falls off of that receptor protein or the concentration drops, then you're going to have fewer receptors bound to it, and then that's going to basically shut off or slow down the signal transduction pathway. So those are all ways that we can make the process more efficient. And then the last thing that we're going to talk about real quick is apoptosis. And what apoptosis is programmed or controlled cell suicide or cell death. And apoptosis often occurs when the cell is damaged or it's not efficient anymore, or there's too much DNA damage. Then a signal will go out to the cell for it to start to die and break down. And so this is a way of cleaning up dead and old cells, but it's also a way of shaping organisms, especially in fetal development. So if we just take a quick look at these pictures, when a fetus is developing, the skin kind of covers the fingers, the bones in your fingers, more like a glove. Sorry, like a mitten. And then as the fetus develops, then apoptosis actually occurs between each of those finger bones, and it shapes the individual fingers of that developing fetus. So apoptosis can help in shaping a fetal development as the fetus grows and develops. The apoptosis will shape those fingers and toes of those developing fetuses. So apoptosis kind of cleans up and straightens up and organizes the cells, and it gets rid of dead and dying cells within the body. So apoptosis is programmed cell death. So just to sum all that up, the signal transduction pathway involves 3 steps: reception, transduction, and response. Reception is when the signaling molecule or ligand attaches onto the receptor. And then transduction is the series of steps that occurs that will lead to a response. And then the response is what happens in the cell as a result of that signal transduction pathway. I hope that was helpful. in understanding signal transduction pathways. And as we move through the unit, we'll talk about different examples of signal transduction pathways and cell signaling when we look at different body systems. So we will take a look at the nervous system, the endocrine system, um, the immune system, and we'll also look at a few examples in plant cells and how they send signals as well. But we'll look at those throughout the unit. I hope this was helpful, and I'll catch you next time.